Pharmapal.
Discovery of VEGFR-2 inhibitors via virtual screening. Hey everyone! Welcome back to our channel. Today, we're diving into the fascinating world of drug discovery, specifically focusing on a key player in cancer treatment, vascular endothelial growth factor receptor 2, or VEGFR-2 for short, was published in Molecular Informatics as Communication. We'll explore how scientists are using cutting-edge technology to find new inhibitors that could potentially revolutionize cancer therapy. So, grab your favorite snack, sit back, and let's get started. First off, let's understand why VEGFR-2 is so important. Cancer is a major health issue worldwide, and it's actually the second leading cause of death in the United States, right after heart disease. As of January 2019, there were nearly 17 million cancer survivors in the US, and that number is expected to rise to over 22 million by 2030. With such a significant impact, researchers are constantly looking for new ways to combat this disease. One of the critical processes in cancer progression is called angiogenesis, which is the formation of new blood vessels. Tumors need nutrients and oxygen to grow, and they achieve this by creating their own blood supply. The problem is, these tumor blood vessels are often leaky and disorganized. This is where VEGF, or vascular endothelial growth factor, comes into play. VEGF binds to VEGFR-2, a receptor on the surface of cells, triggering a signaling pathway that promotes the growth of these blood vessels. Now, here's the kicker: overexpression of VEGF is commonly observed in various cancers, making VEGFR-2 a prime target for new cancer therapies. By inhibiting VEGFR-2, researchers aim to cut off the blood supply to tumors, effectively starving them and preventing their growth. So, how do scientists find new inhibitors for VEGFR-2? One powerful method is called virtual screening. This is where computational tools come into play, allowing researchers to sift through millions of potential compounds quickly. In a recent study, scientists used a ligand-based virtual screening approach with a tool called VROCS, which stands for Rapid Overlay of Chemical Structures. VROCS works on the principle of shape similarity. It helps identify compounds that have a similar shape to known VEGFR-2 inhibitors. In this study, they screened over 2.4 million compounds from the ZINC database and narrowed it down to 19 promising candidates. After identifying these candidates, the next step was to test them in the lab. The researchers purchased these compounds and evaluated their VEGFR-2 inhibitory activity using a specialized assay called Promega's ADP glucokinase assay. This assay measures how well a compound can inhibit the activity of the VEGFR-2 enzyme. Out of the 19 compounds tested, 4 showed significant inhibitory activity, with one compound, labeled as compound 10, exhibiting a moderate inhibitory effect with an IC50 value of 19.3 micromolar. IC50 is a common measure in pharmacology that indicates the concentration of a drug needed to inhibit a biological process by half. But we're not done yet. To better understand how these compounds work, the researchers performed molecular docking studies. This involves simulating how the compounds bind to the VEGFR-2 receptor. They used a tool called FRED for this purpose, which performs a systematic search to find the best fit for the compounds within the receptor's active site. The analysis revealed that compound 10 forms several crucial interactions with the receptor, including hydrogen bonds and hydrophobic interactions. These interactions are essential for the compound's effectiveness as an inhibitor. Now, let's talk about the experimental validation. The selected compounds were purchased from suppliers like Anamine and Cambridge, and then they were put to the test using Promega's ADP glucokinase assay. This assay measures the inhibitory activity of the compounds against VEGFR-2. The results were promising. Out of the 19 compounds tested, 4 showed more than 50% inhibition at a concentration of 50 micromolar. These were compounds 3, 7, 10, and 13. But the excitement doesn't stop there. Compound 10 stood out with a moderate inhibitory activity, boasting an IC50 value of 19.3 micromolar. Now, what does IC50 mean? It's a measure of how much of a substance is needed to inhibit a biological process by half. So, a lower IC50 indicates a more potent compound. Next, the researchers took it a step further by performing molecular docking studies on these compounds. They used the FRED module from OpenEye Scientific software to predict how these compounds bind to the VEGFR-2 receptor. The docking results revealed some interesting interactions for compound 10. For instance, it was found that the oxygen in the nitro group of compound 10 forms a water-mediated hydrogen bond with the side chain of an amino acid called ASPA14. Additionally, the amide group in compound 10 makes hydrogen bonds with another amino acid, gluate85. These interactions are crucial because they help stabilize the binding of the inhibitor to the receptor, enhancing its effectiveness. Moreover, the researchers noted that the pyridyl ring of compound 10 engaged in hydrophobic interactions with several other amino acids in the active site of VEGFR-2. This is significant because such interactions can contribute to the overall binding affinity of the compound, making it a promising candidate for further development. In summary, this study showcases the power of virtual screening and computational drug discovery in identifying potential VEGFR-2 inhibitors. The successful identification of compounds like compound 10 not only adds to our arsenal against cancer but also highlights the importance of understanding the molecular interactions at play. If you found this video informative, don't forget to give it a thumbs up and subscribe for more content on the latest in science and medicine. Have any questions or topics you'd like us to cover next? Drop them in the comments below. Thanks for watching, and we'll see you in the next video.
Pharmapal.